All right. Okay, so I'm Kate Tepper and I'm a PhD student at Macquarie University. And thank you very much to AES for the opportunity to present our lab's research into insect synthetic biology for bioremediation and biomanufacturing. So I'll start out with my acknowledgement of country. So I acknowledge the traditional custodians throughout Australia and I pay my respects to their elders past, present and emerging. So pollution, it's a really big worldwide problem. A review published recently showed that pollution was the leading environmental cause of premature death and disease globally, accounting for 9 million premature deaths per year. It's also listed as a major contributor to species critical conservation status. So it has big implications for species extinction and ecosystems. Another environmental problem is the landfilling of organic waste. So not only could we be using organic waste to generate value added products, but it's also contributing to our climate crisis. So in landfill, organic waste, it's really tightly compacted to reduce the cost of space requirements. And this leads to the anaerobic decomposition of the organic waste into methane, which is the greenhouse gas that's approximately 25 times more potent than carbon dioxide. So we propose to address these challenges using animal synthetic biology. So there's a lot of powerful enzymes that have been evolved in microbes and fungi. And so we could combine the evolutionary strengths of microbes and fungi with the evolutionary strengths of animals, because animals can really rapidly process material, and also their transgene biocontainment is much more effective in animals. So as an example, we could apply this technology for bioremediation and the degradation of pollutants in the environment. So some examples of this is that sea sponges, they're able to filter as much as 14 liters of water every hour. And so we could equip them with microbial enzymes that are able to degrade plastics. And this could be used to manage our microplastic problem in the ocean. Another application for this is that a lot of soil pollutants are taken up by plants. And so we could equip herbivores with enzymes that can degrade these pollutants. And as they eat through the contaminated plants, then you'll remediate the soil and the plants from these pollutants. And the benefit of working with herbivores is that we have really well-established desexing techniques. And so the transgene biocontainment isn't so much of a big issue. Another application for this is that we could engineer fish in waterways to secrete enzymes that can degrade waterway pollutants, or we could engineer the fish with enzymes that can degrade pollutants that bioaccumulate up the food chain, so like methyl mercury and polychlorinated biphenyls. And these pollutants haven't been able to be addressed by bioremediation previously, so it's like a new way that we could manage these pollutants. And then on the other arm of this is that we could use this technology for biomanufacturing. So black soldier flies, they're gaining a lot of traction commercially to rapidly convert a whole different, like a diverse range of organic wastes into low value products such as animal feed and soil amendments. So we could engineer the black soldier flies to convert like a even wider scope of organic wastes into high value biomolecules. So this could include industrial enzymes or commercially relevant lipids. And this technology, it could also be used for bioremediation. So we could prepare lyophilized or like freeze-dried enzymes. And then this could be added to treat industrial wastewater to clean up the pollutants where it might be too toxic for an animal to live in those waterways. And so as proof of concept of these ideas, we first engineered the animal genetic model Drosophila melanogaster. So a lot of the enzymes that we'll be testing, they've evolved for function in their microbial or fungal hosts, and so they might not be functional in a distantly related animal host. And so Drosophila just means we can really rapidly and cheaply test these enzymes, and transgenesis we can perform, like we can outsource that commercially, so someone else can do the transgenesis for us. And then as proof of concept of this, we've demonstrated that we have a functional fungal lacase that could be used for bioremediation and biomanufacturing. So fungal lacases, they're multi-copper oxidative enzymes, so they're able to oxidize and degrade many different pollutants for the purpose of bioremediation. But they also have a lot of different industrial applications. So they can be used in both the pre and post-processing of these industries. But currently, lacases aren't able to be produced economically at scale. And so there's a real technology gap for making large quantities of lacase. So to begin this project, my co-worker, Michael Clark, as a part of his master's program, he generated these genetic constructs. So he picked 2 fungal lacase enzymes, one from Trogii, another one from Polyprispirmalis, codon optimized them for expression in Drosophila. And then these 2 enzymes, they're both secreted in their fungal hosts. And so we replaced the fungal signal peptide for a Drosophila signal peptide. And then to drive the expression of these enzymes, we chose the short alpha tubulin promoter. And this is because it is expressed across all tissues. So right now, we're not really sure what the optimal expression patterning would be. And so by expressing it everywhere, we sort of increase our chances that it will be functional somewhere. And then this promoter also provides mid-level expression. So we weren't sure whether these enzymes might be toxic if they're overexpressed. So by only expressing them at a mid-level just means that, they're less likely to be toxic in their host. So for our first experiments, we showed that the Trametes trogii lacase was functional in the flies. So one of the benefits of working with lacase is that there's a really simple chromogenic enzyme activity assay that you can do. And this is using the chromogenic substrate ABTS. So as the lacase oxidizes the ABTS, it turns from colorless to a blue-green color with an absorbance maxima of 420 nanometers. And so the Drosophila melanogaster that contained the Trametes trogii lacase, when I incubated these fly lysates with ABTS, we saw a significant increase in ABTS oxidation compared to the wild type lysates, but we didn't see any activity in the Drosophila melanogaster that had the Polyprispirmalis lacase. And so we just focused on working with the Trametes trogii lacase for the future experiments. So then we wanted to show whether this activity would work in vivo. And so what we did was we set the ABTS with the fly diet and then incubated the flies in the diet. And as the larvae hatch and then they eat through the media, then they'll secrete that enzyme and be able to oxidize the ABTS. And so as you can see here, the flies that had that lacase could oxidize the ABTS, and you see this blue-green color, but we didn't see any oxidization in the wild type flies. So now we know that our enzymes are functional. We next moved on to characterizing whether it could degrade an environmentally relevant pollutant. And so to do this, we used the environmental pollutant Bisphenol A. So this is a compound that's used to make plastics. And it's been shown to leach from plastics and disrupt our endocrine system. And so when I incubated the fly lysates with BPA, we saw a significant reduction in BPA concentration compared to the wild type lysates and the no-fly control, which contains all the assay components except for the fly lysate. And then from this data, I've just shown here the LCMS extracted ion chromatograms that we would expect for the BPA peak. And as you can see, the BPA peak is much smaller for the samples that contain that fly lacase compared to the wild type and the no-fly control. But when we went to test this in vivo, it didn't appear that it could degrade BPA compared to the wild type flies. But we did see a significant reduction in BPA compared to the no-fly control. And we think this might be because the flies might actually contain endogenous detoxification pathways to be able to degrade BPA. And this has been characterized in mammals, so it's possible that it's also doing this for the flies. The other possibility is that BPA is just extracted much more readily from just media than maybe the BPA absorbs much more readily to the fly larvae, so we can't extract as much. So to begin troubleshooting this, we also incubated the media with the redox mediator ABTS, which has been shown to assist laccase activity previously. And so we showed that when we incubated the media, when we added BPA and ABTS in the media, we saw a significant reduction in BPA concentration in the vials that contained the flies with the laccase transgene. And so this is like a really exciting result. It represents the first example of an engineered animal remediating a xenobiotic pollutant and warrants further research into other animals that might be more applicable to remediation in the field, and also looking into additional enzymes that are capable of degrading pollutants. And so the next part of this project was to test if we could prototype insect bioproduction. So we wanted to prepare a freeze-dried laccase from the adult flies, and this is means that like it could be readily stored and transported for downstream industrial utility. And so when I did an enzyme activity assay comparing the crude freeze-dried laccase prepared from adult flies to a commercially available purified laccase from Sigma. So yeah, I guess this result might not look like much, but yeah, I am comparing the total, all the other proteins that might be in the flies as well. So it's not a purified laccase to the purified laccase that's available from Sigma. So it's likely with, if we optimized our constructs, like a genetic expression for this purpose, it could be improved much more. And then we want to show whether this had downstream industrial utility to clean up industrial wastewater. And so BPA is an industrial pollutant. And so when we incubated the freeze-dried fly powder with unbuffered water and copper, we showed that the BPA concentration could be reduced by 96% after 90 hours. And this was compared to the wild type and no fly control, which we didn't see any degradation. And then another application is cleaning up the industrial dye indigo carmine. So this is the dye that's used to make blue jeans by textile industries. And so if it's released into the environment, it can disrupt photosynthesis, like photosynthetic species in waterways. So it can impact ecosystems. And so yeah, here I'm showing that we can decolorize the indigo carmine dye compared to the controls. And then when we add the indigo carmine with the freeze-dried laccase in unbuffered water with copper, we saw a almost complete decolorization of that dye with the freeze-dried fly laccase. So in summary, we were able to successfully engineer Drosophila melanogaster to express a laccase from the fungus Trogii Trogii. And then we were able to prototype animal bioremediation. So the transgenic flies could degrade BPA in vitro and in vivo when the redox mediator ABTS was present. And then we were able to prototype insect bioproduction. So we could make a freeze-dried laccase from the adult flies. And this could be used for industrial, to clean up industrial wastewater that might contain BPA or indigo carmine. And thank you to everyone in my team and our collaborators and our funding and support to help make this research happen.
Great, thank you, Kate. We are out of time, so we don't have a moment for questions, but I'm sure you can message Kate for questions later on.